Namaskar dosto, welcome to channel Toxicology on Board. Today in this video we'll be discussing another important DABT topic, that is methyl mercury, dimethyl arsine, dimethyl selenide, methylation, metabolic pathway. So let's directly dive into the question. So this question was taken from ABT recertification examination May 12, 2000. Okay, the question is, some elements can be methylated by metabolic pathways in vertebrate species or by microbes in water, sediment or soil. Important products of biomethylation include. So what we need to identify is which of the following elements will undergo biomethylation. Pathway might be anything. Different. It is different in vertebrates. It is different in microbes in water, sediment or soil. Okay. So the first option is option A, trimethyl tin, triethyl lead, methyl mercury. Option B is methyl mercury, dimethyl arsine, dimethyl selenide. Option C is trimethyl tin, methyl mercury, dimethyl arsine. The option D is only methyl mercury. Option E is methyl mercury, dimethyl cadmium, tetraethyl lead. The right answer will be in the green text, that is methyl mercury, dimethyl arsine and dimethyl selenide. Okay, so important, I will not discuss about other options unlike previous videos, no. I used to discuss about other options also. Now I will not discuss about trimethyl tin. I will not discuss about tetraethyl lead. Okay, because the option itself is having three things. So in this video we have to discuss about methyl mercury. We have to discuss about dimethyl arsine, dimethyl selenide. Another important thing you have to remember, whenever you are preparing for the exam, no, you will come across this methyl mercury, its biomagnification, biomethylation. So when you look into the options, no, you will think that option D is only the right thing. Okay, but in addition to option D, which is methyl mercury, other two options like dimethyl arsine as well as dimethyl selenide, these two are also undergo biomethylation. But tetraethyl tin as well as tetraethyl lead, dimethyl cadmium, these will not undergo biomethylation. Okay, you have to remember. So you have to choose the answer. This in this question, no, it is a tricky question. He's trying to confuse you. Our classical example will remember only methyl mercury. So when we see option D, we'll think that D is only right. But the option is B. In addition to methyl mercury, other two elements, that is dimethyl arsine as well as dimethyl selenide. So selenium, arsine and mercury, all these three things will undergo biomethylation. So we'll discuss in detail about each and everything. So first one we'll discuss dimethyl, sorry, methyl mercury. In my channel I have already uploaded a lot of videos with respect to methyl mercury magnification. Directly from the Clayson also I have given some notes in that videos. Go back and check. Since we are discussing this question here, little bit of about methyl mercury and biomagnification, we'll get ourselves introduced to the concept of this bio amplification, biomagnification, and we will see what are the definitions also. Okay, so methyl mercury, it will undergo biomethylation, is the microbial conversion of this inorganic mercury. Mercury will be Hg0 state, it will be there. Inorganic state, inorganic mercury. This inorganic mercury will convert into organic mercury, into highly toxic organic methyl mercury. Okay, here methyl organic is the CH3Hg. Okay, here you can see CH3Hg. Primarily in the aquatic sediments, that is in the water bodies, a process crucial to the mercury cycle. So in the environment, mercury undergoes cycle, no, that for this mercury cycle, this is a primary first important step that leads to its accumulation and biomagnification up the food web. So in the food chain, no, as you go above the food chain, the concentration of this particular methyl mercury, it keeps increasing over the food chain, posing significant neurotoxicity risk. So those people who are high above the food chain, no, those will have CNS toxicity will be there. Risk to humans and as well as wildlife, higher animals, especially via fish consumption with sulfate reducing bacteria and methanogens being the key players. So these are the important. So sulfate reducing bacteria are the one, and methanogen, and there are certain physical conditions, pH conditions also, which are favorable for the transformation of this methyl mercury, inorganic mercury to methyl mercury. So let's see what is biomethylation. Biomethylation is nothing but it's a biogeochemical process where microorganisms add a methyl group. So to this inorganic mercury, CH3 group is added to inorganic mercury, Hg plus 2. Okay, and the key product are methyl mercury is it is a potent neurotoxin that readily crosses the biological membranes. Okay, phospholipid bilayer membrane, it will be able to pass through the cell membrane, including blood-brain barrier as well as a placental barrier. That is the reason this methyl mercury it is causing fetal toxicity, and it is crossing the blood barrier. That's why it is causing CNS toxicity. Okay, because of its lipophilic nature. Okay, what is the main important steps which are involved in the process are inorganic mercury input, mercury from these natural and pollution sources. So there are certain factories where the mercury is leached into the nearby water bodies or into the soil, or there is volcanic eruptions are there, or there is any mining is taking place where this mercury is leaching into the soil. Okay, so from natural or pollution sources like coal burning, gold mining enters into the aquatic environment. So nearby any water bodies are there, in that mercury will be released. Microbial conversion. Now the bacteria, particularly sulfate reducing bacteria, SRB, that you have to remember, and methanogens in anoxic oxygen poor sediments, they will use these enzymes to methylate. So CH3 group is added to the inorganic mercury, methylate inorganic mercury. So methyl group sources are, methyl group is from where this methyl group it is coming? It is coming from methylcobalamin, which is a form of vitamin B12 or other methylated compounds. So what are the environmental factors which are favorable for the transformation of this change is pH, organic matter, temperature, oxygen levels influence methylation rate. Whether the methylation taking place at a faster rate or slower rate, all these things are dependent on these environmental factors. Okay, so anoxic oxygen poor sediments. Okay, then let's see in detail some more additional points with respect to why this bioaccumulation or biomagnification is important. Bioaccumulation, methyl mercury builds up in individual organisms over a time. As I told you, as the food chain increases, the concentration of that methyl mercury keeps on increasing up the food chain. Concentration increases significantly at higher trophic levels, that is small fish, larger predatory fish, followed by humans. Human health, consumption of this particular contaminated fish is the main exposure route, leading to severe neurological damage, especially in the developing fetus, because it is able to cross the placental barrier. That's why it is causing fetal toxicity, teratogenicity, right? Then I have uploaded one video, as I told you, in my channel with respect to Chizo Minamata Bay disease. Also, please go read Minamata Bay incident which has occurred in the Japan. So that is particularly specific one video on methyl mercury only I have uploaded. Please go read that also. Okay. And the key, what are the key microbes which are involved in this particular biomethylation process is sulfate reducing bacteria, SRB. They're the major contributors in anaerobic sediments. D-sulfomicrobium escabenaceae. Methanogens, what are those methanogens? Also significant producers in anaerobic conditions. Aerobic bacteria. Recent research shows that some aerobic bacteria can also contribute to this methyl mercury production. And biomethylation, transformation of relatively, first of all, this inorganic metal, no, it is immobile. It is not able to transfer in the either in the food or in the water or in the soil. But this immobile metal, it is transformed into highly mobile, dangerous organic toxin, which is nothing but methyl mercury, making mercury pollution a serious public health and ecological problem. So please read about this conversion here I kept, and these are the sulfate reducing bacteria and methanogens which are involved in the biomethylation process. Okay, so this is a biomagnification food chain you can see. Also read Klesenka important chapters, and diagram is also very clearly given. Now we will see second option, that is dimethyl arsine, arsenic from it is coming. It is a volatile, highly toxic intermediate in biological methylation, biomethylation of inorganic arsenic. So similarly like inorganic mercury, this inorganic arsenic is also converted into dimethyl arsine, organic arsine. compounds by various microorganisms. What are those? Bacteria, fungi, and archaea. Okay, the process is a key part of the global arsenic biogeochemical cycle. Similarly, like mercury cycle in the environment, similarly you have global arsenic biogeochemical cycle, often considered as detoxification mechanism. So the elemental form of arsine, which is highly toxic, it is converted into less toxic form, I believe. So biomethylation typically proceeds by a challenger pathway. A series of alternating reduction and methylation steps are involved. Okay, and by the action of this particular enzyme, what is that enzyme? S-adenosyl L-methionine. This particular enzyme will be acted upon this process as the primary methyl donor. This S-adenosyl methionine will facilitate the methyl group to this particular arsenic, and thiol compounds like glutathione, GSH. Okay, so what are the process? We'll see uptake and reduction. As I told you, inorganic arsenate, which is in fifth oxidation state, is taken up by the organism and reduced to arsenite, As plus 3 oxidation state. Monomethylation, As3 is there, it is oxidatively methylated by this particular S-adenosyl methionine SAM enzyme, which will facilitate the methyl group. This As3 methyl transferase by this enzyme, this arsenic M2As3MT enzyme to form monomethyl arsenic acid, MMA5. So here you can see the diagrams in the pictorial form. You can see how the fifth state of oxidation converted to third state by action of this MMA, TMA, DMA, ASH3, arsine gas is formed. So like this, it is very difficult to remember for all this oxidation state. Just to get yourself introduced to this concept. Okay, is dimethylation, MMA5 is there, it is reduced to this monomethyl arsenious acid, which is then methylated to form dimethyl arsenic acid, DMA5. Okay, and dimethyl arsine formation further reduced to trivalent form. Okay, the dimethyl arsenious acid, DMA3, which can then be converted into volatile gas dimethyl arsine. Important thing that you can remember from this slide is arsine gas formation is important, and what are the enzymes uptake reduction, how the various oxidation state, everything you can't remember, but at least up to arsine gas you try to remember. Okay, that was about some details about the arsine. We'll see what are the important significance and toxicity with respect to this dimethyl arsine. Dimethyl arsine along with trimethyl arsine, TMA, it is a volatile species that can escape from water bodies or soil surfaces into the atmosphere, contributing significantly to this, as I told you, global cycling and transport of this arsenic. And what are the important toxicities which is caused by this particular dimethyl arsine is its pentavalent methylated species MMA5 or DMA5. They are less toxic. Fifth form of oxidative state is less toxic than inorganic arsenic. Some trivalent intermediates are there like MMA3, DMA3. Previous slide you have seen, they are highly toxic. Fifth oxidation state say comparatively third one is highly toxic, and fifth one is less toxic when compared to inorganic arsenic. Dimethyl arsenic gas is also extremely toxic. So one question on dimethyl arsine, ASH3, something like that they will ask you, and it is potential genotoxic. It causes DNA damage. So coming to bioremediation, the ability of certain microbes to convert this arsenic into less toxic volatile forms, that is dimethyl arsine, has a potential applications in this bioremediation of this arsenic contaminated environment. For example, there is a lake or there is a water body or some particular landfill is there which is contaminated with arsenic. By using this particular microbes or this thing, this bioremediation process, we can make that one less toxic. Okay, dimethyl arsine, it is a critical transient link in the complex biological pathway that organisms uses to process or eliminate this inorganic toxin. Arsine is a which is highly toxic from the system or from that particular environment. Okay, so that was about regarding second option, that is dimethyl arsine. Now we'll try to read about another third one, that is nothing but dimethyl selenide, which is coming from the metal selenium. It is a volatile gas produced during biomethylation of selenium. So first one we read about mercury, second one we read about arsenic, third one we are talking about selenium. Okay, a process where microorganisms, bacteria, fungi, they'll convert this inorganic selenium into this organic form. Always inorganic mercury to organic, inorganic arsenic to this one. Okay, volatile form often as a detoxification mechanism. So they are trying to make the organic form, inorganic form of the metal, which is highly toxic, to a less organic toxic form. Okay, releasing into the atmosphere as a less hazardous smelly compound, crucially for selenium cycling and potential bioremediation. So there is certain area which is contaminated with selenium. By using this bioremediation process, you can decrease the toxicity of this particular selenium in that particular area. So biomethylation process, what are the microbes which are involved? Similarly, like bacteria, fungi in the soil, sediments, water convert toxic inorganic selenium like selenide or selenate into volatile organic compounds. And coming to detoxification, this methylation is a key detoxification process for the microbes, reducing the selenium toxicity. And product DMSC is a major product, dimethyl diselenide and dimethyl selenium are also formed by the byproduct, but they are not the more toxic products. Okay, and what is the fate? The environmental fate is that once this dimethyl selenide is released into the environment, no, selenium can move through the atmosphere, eventually being dispersed and deposited elsewhere, effectively removing it from the contaminated sites. Okay, so this is a schematic diagram of the selenium geochemistry in different earth components you can see, and these are the things which are formed: dimethyl diselenide, natural organic matter, dimethyl selenide, organic matter, etc. You can go through this particular diagram. And what are the significance and implications of this dimethyl selenide is that biomethylation is a major driver for selenium movement in the environment. Okay, enhancing DMSC production is a promising method to remove selenium from the contaminated agriculture drainage. With respect to selenium, also you read about selenium contaminated some grass or plants are there. If the grazing animals, livestock animals are consuming that particular low, they will have some kind of toxicity is there. On that, I have already uploaded one video in my channel. So please go through that one. Okay, selenium, how it is affecting the grazing animals, the toxicity, livestock. Okay. Then next, less toxic. DMSC is significantly less toxic than its inorganic precursors. Okay, making microbial volatilization is a useful remediation strategy. Microorganisms uses biomethylation to off-gas selenium as DMSC, transforming a potential environmental toxin into a mobile, less dangerous form, offering a natural way to manage the selenium pollution. Okay, so hope from this video you were able to understand about biomethylation, biomagnification. Apart from methyl mercury, also remember about selenium as well as arsenic, which also undergoes this biomethylation. Saving the environment and landfill regions with such kind of processes is very, very important. Hope from this video you will able to learn about these particular things. Thank you so much. Be curious. For any questions, please write a mail to arya.priyanka18@gmail.com. Like, subscribe and follow my YouTube channel Toxicology on Board. All the best for your exam. Thank you.